All right, so this video is going to go over all things important from Unit 2.3. I'm going to take the cell cycle, break it down into all of its pieces, and discuss in detail what's happening during each part of the cell cycle. So in the life cycle of a cell, it's going to be spending most of its time in a phase called interphase. Interphase is the time where the cell is growing, being a cell, making its proteins, and preparing for cell division if necessary. Then the other phase is the M phase, and that's where we're actually dividing the cell into two different cells. So let's start at the beginning of cell growth, which is the first part of interphase. It includes a phase that we refer to as G1. It's this growth and normal, it says metabolic roles. Well, those metabolic roles are building proteins. So if we look at building proteins, the very first part is that we get instructions from the DNA in the nucleus on how to make those proteins. The ribosomes are going to decode that information and they're going to be able to actually create those proteins. Once the proteins are made, they're going to move into what is called the endoplasmic reticulum, or the ER. So those newly made proteins are going to move from the ribosome through the ER, and they're going to travel through that ER. The ER is going to place them into a vesicle potentially, if they're larger, and move them through the cytoplasm out to the Golgi apparatus. In the Golgi apparatus, they can then be packaged and organized and labeled, and those proteins can either be released into the cell to do a job, or they can be released out of the cell to communicate or work with neighboring cells. So here we can see from the Golgi apparatus that protein coming in as a vesicle and then moving out into the other part of the cell or outside of the cell after it has been repackaged, reorganized, and labeled. So that's what's happening during G1. Some cells stay in G1 for a long time, some for not very long. It just depends. Once a cell starts to get too large, it's not very efficient. So at that point, it's going to start to signal itself to, hey, we need to move on and move into the next phase and get ready for cell division. So at the end of G1, there's a feedback signal that comes in the form of a cyclin that tells that cell, hey, let's move on to the next phase of interphase, which is the S phase. S stands for synthesis. So synthesis means to create. So what we're going to create is a copy of every single chromosome. When we make those copies of the chromosomes, we're going to hold those identical copies or sister chromatids together at what's called the centromere. Those two identical sister chromatids are held together at the centromere so that we don't lose those copies. So once the DNA has been copied, the rest of the cell starts to prepare for cell division, and that's what happens during G2. So the cell will grow a little bit more. It may get some more cytoplasm, it may get additional organelles, but we're really going to get to that point where we're ready to divide. At the end of G2, there's another feedback signal that's going to say, hey, did we replicate the DNA during the S phase? Do we have enough organelles? Do we have enough cytoplasm to divide? If the answer is yes, then the cell's going to move into the M phase, or into mitosis, which is the actual division of the nucleus and all of that important DNA. So the first phase of mitosis then is going to be prophase. And in prophase, so we have a diagram here, the chromosomes are actually going to condense and they're going to be visible for the first time. We can actually see each individual set of chromosomes or sister chromatids. We also see spindle fibers start to come from the centromere, and they're going to help move things around the cell. And the last part of this prophase is that nuclear envelope, or the nucleus itself, starts to break down. So that DNA is going to spread out a little bit and not be as packed as tightly as it was when the cell was growing and developing. Then we are going to kind of not talk too much about this pro-metaphase. We're going to move straight to metaphase. So in metaphase, we're going to see those sister chromatids lining up in the middle of our cell. So the centromeres that are holding those two identical sister chromatids together are going to attach to a spindle fiber. When they do that, we can now have our third feedback of our cell cycle. And that third feedback signal that we get is, do we really have two copies of every chromosome lined up in the center of our cell? So this is the last opportunity for your cell to make sure it has all the pieces and parts that it needs to create two identical cells. So after metaphase, we move on to anaphase. And anaphase is where that centromere gets split and we have one copy of DNA pulled to one side of the cell and the other copy pulled to the other side. So now our two identical copies of DNA are separated to opposite ends of the cell. This happens because the spindles are going to be pulling those copies of chromosomes apart. And then the very last part of the M phase is telophase. So during telophase, a new nucleus is going to form around each set of chromosomes. So we're going to end up with two nuclei, but they're still sharing their cytoplasm and organelles. So that's going to get us through the mitosis phase. Then the last part of that M phase or that division phase is cytokinesis. Cytokinesis looks different in a plant cell compared to an animal cell. So cytokinesis is when the cytoplasm and organelles finally divide into two completely separate cells. When cytoplasm occurs in an animal cell, we're going to pinch that cell membrane together. And when we pinch the membrane, the cytoplasm gets squeezed into two separate halves. With cytokinesis in a plant cell, because of the rigid cell wall, we're going to build a cell plate in the center to separate into two separate cells. Once it's separated into two separate cells, those two cells will then go right back into growing and dividing or growing and making proteins just like they started out. Now, I talked about three checkpoints throughout the cell cycle. The first one was at G1 telling us, hey, we need to divide. We're getting inefficient. We're too large. We're not able to make our proteins. Something's not happening right, and we need to divide. The other one is at the end of G2 to make sure that we have our copies of DNA and all of the pieces and parts that we need for cell division. And the last feedback occurs during metaphase, where we make sure that we have those two copies of sister chromatids lined up in the middle. If at any point that feedback does not communicate properly to the cell or the cell does not respond properly, we can have out of control cell growth, which can lead to tumors and cancer and uncontrollable cell growth. So those are the key points. Go ahead and work on your key point review worksheet. Make sure you do your quizzes to a 90% accuracy that has been posted, and you will be ready for your retake. So you will turn in your test authorization, retake authorization, and your worksheet to your teacher so that you can arrange to take your retake. Good luck.